Hello, in this video, I'm going to talk about some introductory nervous system topics. So first, if we're going to name all of the functions of the nervous system, absolutely everything the nervous system does fits into either the category of sensory, motor, or integration. Okay, so to sum it up simply, what does the nervous system do? Sensory, motor, or integration. Another word for integration in this context is also association. So you'll see both words used to mean the same thing, so integration or association. So sensory function, we're talking about all of the incoming information that is coming from um, information that we're collecting about things outside the body and information about things happening inside the body. So we're collecting all of this information about what's happening around and inside the body and sending all of that into the central nervous system. Central nervous system just being the brain and spinal cord. So the brain and spinal cord are receiving all of the information that we're gathering about everything that's going on. Motor function is all of the outgoing information from the central nervous system. So the brain and spinal cord are sending out instructions out to the body to tell muscles to contract and to get contents to move along in the intestines and to tell glands to secrete. So those are all motor functions. So all of the outgoing instructions sent from the brain and spinal cord out to instruct the body what to do, that's all motor function. And then finally, integration, again, also called association, includes everything else. It's all of the processing of the sensory and motor information that we do. So it's like we take in sensory information, but that's not enough. We can't just take in that raw information. We have to then interpret what it is, what it means, and then maybe come up with a motor response because of it. So like if you smell something, okay, so you smell something in the air, we'll take in that sensory information that gets sent into the brain, but that smell means nothing. That odor that you sensed in the air means nothing until your brain has interpreted what that means. It will compare that sensory information against past experiences and see, have we smelled this thing before? And if we have, what was it last time? And that helps you interpret what it is that you're smelling in the air. So you might smell something, compare it to what you've smelled before, and realize that somebody's baking cookies. It smells just like, chocolate chip cookies that you used to bake a long time ago or something. Okay, so you compare it to your past experiences to interpret what is happening. And then you smell those cookies and maybe you want to go to the kitchen and get one. And so then that's where your brain will send out a motor command or a whole series of motor commands to cause you to walk and move to the kitchen to go get a cookie. Okay, so all of that happens in between the sensory coming in and the motor going out, everything that happened in between was all integration or association. So it's all that processing that happens between the sensory and the motor. All higher level thinking, so memory, planning, emotions, and all other aspects of cognition, that all is in the group called integration. So again, everything the nervous system does is either sensory, motor, or integration, and we could sit here and list things all day long that fit into each of these categories. Okay, neurons are the cells that provide most of the unique functions of the nervous system. That is because they are electrically excitable. So just like muscle cells, they have the ability to generate an action potential in response to a stimulus. So that stimulus could be a sensory stimulus. So if this is a sensory receptor, then that stimulus would be some kind of sensory stimulus. So that could be like smell, pressure, vibration, light. So there are many, many different sensory stimuli that different sensory receptors are sensitive to, and that's what causes them to generate an action potential. But also, we also refer to it as a stimulus when we have one neuron that is communicating with the next neuron. So when there's a first neuron that is secreting a neurotransmitter into the space between the two neurons to stimulate the next neuron to also generate an action potential. That neurotransmitter coming from the first neuron is a stimulus. So if there is enough of that stimulus, so if that first neuron secretes enough neurotransmitter, or maybe there are multiple neurons secreting a neurotransmitter, and so if there's enough of that neurotransmitter interacting with the neuron that's receiving the message, then we would say it reached threshold and then generated its own action potential. Okay, so that's another example of how we use that word stimulus. So stimulus could be a sensory stimulus, or it could simply mean that there was enough neurotransmitter to stimulate an action potential in a neuron. So an action potential, I just use that word several times. We use that interchangeably with the word impulse. It's an electrical signal generated by a neuron that travels down the axon to send a signal to another neuron or to an effector. So like we see in the picture here, this is a polarized neuron, meaning that we have a separation of charges. It's polarized. It's positive outside, negative inside. So this is a polarized neuron, meaning that it's ready and waiting in case there is a sufficient stimulus to cause the neuron to reach threshold. Then it will open gates that allow the positive ions to flow in. So our negative charge inside becomes positive as those ions sweep down the length of the axon. So as those positive ions sweep in to the axon, it causes more and more channels to open, or gates to open, so that more and more positive ions can flow in until the whole thing is positive. And that is what the action potential is. So it's the traveling of that positive charge down the length of the axon. So then when the action potential is over, then the cell is going to pump out those positive ions very quickly and repolarize, so that we go back to this resting state where it's negative charge inside and positive charge outside, so that it's ready to generate the next action potential as soon as possible. Okay, so as I mentioned, a stimulus is any change in the internal or external environment that may generate an action potential. So that can mean a lot of things, as I mentioned. That can mean all kinds of sensory stimuli that we might be detecting inside or outside of the body. But also a stimulus would be a change in the amount of neurotransmitters that would be detected by that neuron. So as long as there's a sufficient change that it would cause an action potential, that would be considered a stimulus. An effector is a structure that responds to motor commands to have effects in the body. So that's a very short list. The only effectors in the body are muscles and glands. It could be any of our three types of muscle tissue, and it could be endocrine or exocrine glands, but those are the only things in the body that have the ability to respond to motor commands sent by the central nervous system. Okay, so the brain and spinal cord are sending out motor commands out to the effectors of the body. And those effectors are always only skeletal muscle, cardiac muscle, smooth muscle, endocrine glands, or exocrine glands. So those are the only things in the body that are responding to our motor commands coming down from the central nervous system. Okay, neuroglia. This is a whole category of cells that nourish, protect, and support neurons to enable them to function. They significantly outnumber neurons and are not electrically excitable and have many, many functions, such as forming myelin sheaths. Okay, so if we look at all of the cells in the nervous system, all of them are either neurons or neuroglia. And there are lots of different kinds of neurons, and there are different kinds of neuroglia, but all of the cells in the nervous system fit into those two categories, neurons and neuroglia. As we've talked about, neurons are electrically excitable and they are able to generate an action potential. Neuroglia are not. They have a supporting role, and there are many, many more of them compared to how many neurons there are. So you could sort of think about it as the neurons being like the race car drivers. They're the ones that are really doing or having the effect that we need in the nervous system. But the race car driver or the neuron can't do its job, or at least not well, without having a pit crew. So the neuroglia are like the pit crew. There are many more neuroglia to support every neuron. So they're performing all kinds of supportive features or functions, like they're protecting, they're supplying nourishment, they're protecting them from like toxins and things in the blood. Like it's neuroglia that form the blood-brain barrier, for example. It's neuroglia that form myelin sheaths, which we'll talk about in a minute. So there are many more neuroglia that are all performing different functions to protect and support the neurons so that they can do their job effectively and efficiently. So a myelin sheath is a structure that surrounds the axon of a neuron to speed the transmission of the action potential by insulating the axon. So if you think about the axon of the neuron as being like a wire, which is really what's going on, we're sending an electrical impulse down the length of a wire. The myelin sheath is the insulation of that wire. So first and foremost, the function that the myelin sheath provides is that it speeds the rate of travel of that action potential. That is the first and foremost function that is most important that they provide. In addition to that, they're wrapping around the length of the axon, so it also provides protection against injury of the axon. So a myelinated axon carries an impulse significantly faster than an unmyelinated axon, which is an important advantage, especially in very long axons. Okay, so not all axons are myelinated. Some are myelinated and some are not. Most are myelinated, and especially the longer the neuron is, or the axon is, the more likely it is to be myelinated. The larger and kind of thicker the axon is, the more likely it is to be myelinated. It's really the very smallest axons that are unmyelinated, mostly because the axons are too small to support the structure of a myelin sheath surrounding it. So in the very small and or very short axons, there isn't myelination there. But in pretty much all the other axons, we're going to have myelination because it significantly speeds the rate of travel. So if we have an axon that could be a foot long or 2 feet long or longer sometimes, you want there to be very, very fast transmission of the action potential, rather than if it's not myelinated, it takes a very long time for that action potential to travel all the way from one end to the other. That's because the action potential actually travels through the myelination itself. So the myelination those are actually neuroglial cells that are wrapping themselves around the axon. So those cells are full of intracellular fluid, just like all the other cells in our body. So they're full of fluid. And if you think about, like, if you had a plugged-in toaster and you walked over to a swimming pool and dropped it in on one corner, the whole swimming pool would immediately be electrified. And so it's kind of the same idea. These are cells that are full of intracellular fluid. And so when the action potential, which is an electrical charge, reaches one end, it immediately electrifies that whole part of the myelin. And so it essentially causes that charge to hop all the way to the other end of that section of the myelin. And so because of that, it makes it look like the charge is hopping between little breaks in the myelin, because it's going from swimming pool to swimming pool, essentially causing the whole thing to electrify. So it causes the charge to move from one end of the axon to the other much, much, much more quickly than if there was no myelin and the electrical charge was just slowly kind of traveling down the myelin without being able to hop much more quickly. Okay, a tract is a bundle of axons or projections of neurons that are only located in the central nervous system. So if it's a bundle of axons in the central nervous system, it is a tract. If it's a bundle of axons in the peripheral nervous system, it's a nerve, which we'll talk about in the next slide. Okay, so a bundle of axons in the central nervous system. There are many ascending and descending tracts traveling along the spinal cord and brainstem. So we're saying that these bundles of axons are forming these sort of superhighways that are either going up, so going up the spinal cord towards the brain, or coming from the brain, coming back down, so ascending or descending tracts. So what we're seeing in this picture here is a cross-section of the spinal cord that's showing kind of a map of the tracts in the spinal cord. So the blue tracts are carrying information up to the brain, and the red tracts are carrying motor commands down from the brain out to the body. Okay, a nerve, again, is similar. It's a bundle of axons, but here we're talking about in the peripheral nervous system. So if it's in the brain or spinal cord, it's a tract. If it's anywhere outside of that, it's a nerve. So by definition, there is no such thing as a nerve in the brain or spinal cord, and there are no tracts outside of the brain or spinal cord. So we name our nerves depending on where they emerge from the central nervous system. So we have 12 pairs of cranial nerves, and they're named that because they all exit the brainstem. And actually, cranial nerve 1 and 2 actually exit from the cerebrum, but in any case, they're all leaving from the brain. Okay, so we have 12 pairs that are leaving from the brain. Most of them from, 10 out of 12 are leaving from the brainstem. Cranial nerves 1 and 2 leaving from the cerebrum. Then we have a pair of spinal nerves exiting at each level of the spine. So we have 8 pairs in the cervical spine, and that's because we have 7 cervical vertebrae. We have 1 pair that exits just above C1, and then a pair that exits below each of the 7 vertebrae. So that's 8 pairs altogether. Then we have 12 pairs in the thoracic, 1 below each thoracic vertebra, 5 in the lumbar, 1 below each lumbar vertebrae. And then we have 5 pairs that exit at the sacral level and 1 pair at the coccygeal level. So altogether, we have 31 pairs of spinal nerves that are exiting all the way as we go down the spinal cord. So many of those nerves that exit the spinal cord form plexuses. So a plexus is a network of nerves. We also use that term to describe blood vessels, which is why I made that note here. So plexus could be of nerves or of blood vessels. Here, of course, we're talking about networks of nerves. So we have 4 major nerve plexuses that exit the spinal cord. So the spinal nerves exit individually at each level, but once they do, they will split off and join together and split off and join together so that we have axons that are traveling in lots of different paths instead of just taking the path of one individual spinal nerve. We have opportunities where it's basically like highways that are crossing paths so that axons can travel down different paths to go different places. And so that's really what a plexus is in this context. So the cervical plexus supplies the posterior head, neck, upper shoulders, and diaphragm. And those are coming from the nerves that are coming off at level C1 through C5. The brachial plexus is just inferior to that, and it's named that because it's going out to the arm. It's going out to the upper extremity. So it supplies some neck and shoulder muscles in the entire upper extremity. So that's coming out at levels C5 through T1. Then everything in between, so T2 to T11, those nerves do not form plexuses. Okay, so we commonly call those intercostal nerves, and they just stay individual and they're staying local in the trunk. Okay, so T2 through T11 are not part of a plexus. Then we get to the lumbar plexus, and that's T12 through L4, supplies part of the lower extremity, the abdominal wall, and the external genitals. And then below that, we get to the sacral plexus that supplies most of the lower extremity, the buttocks, and the perineum. Okay, and so that sacral plexus, that's L4 to S4. Okay, chemical synapse. An example of a chemical synapse is at the neuromuscular junction. So this picture probably looks familiar since we've talked about the neuromuscular junction in past videos. Um, and so what we're seeing is a neuron that is secreting a neurotransmitter. So it's a chemical, hence its name, chemical synapse. So it's secreting a chemical, a neurotransmitter, out into the synaptic cleft. That's the space between the neuron sending the signal and whatever is receiving the signal. And then something is receiving that signal on the other side. So in the neuromuscular junction, we're looking at a motor neuron who is secreting acetylcholine that travels across the synaptic cleft and causes contraction. It's received by that muscle cell on the other side and causes an action potential and a contraction in the muscle. In this picture, we're seeing a presynaptic neuron that is sending a signal to a postsynaptic neuron. So those words, presynaptic and postsynaptic, are really just referring to, is it the neuron sending the signal or the neuron receiving the signal? So before or after where the synapse is occurring. Okay, so in this case, we have a neuron sending a signal to another neuron, but the process is exactly the same. So we're secreting a neurotransmitter. It could be acetylcholine, but there are a number of others. There are many, many others. So it could be some other neurotransmitter that is then acting as the stimulus to cause an action potential to generate in the postsynaptic neuron or in the effector, depending on what we're looking at. Okay, so a chemical synapse is the transmission of an action potential from one cell to another. It's when a neuron sends a chemical signal to another neuron or to an effector. So both would be a chemical synapse, whether we're going neuron to neuron or neuron to effector. Neuron releases a neurotransmitter, could be acetylcholine or many others, that travels across the synaptic cleft to bind to receptors on the other neuron or effector cell. And importantly, chemical synapses make up about 90% of all of the synapses in the body. Okay, so most of the synapses in the body are chemical synapses. The other 10% or so are electrical synapses. So in an electrical synapse, we have the transmission of an action potential from one cell to another via gap junctions. So gap junctions are what we see in the picture here. Those are those gray things that are connecting the two cells and forming sort of a tunnel, making a channel. Okay, so they're tunnels that allow the ions of an action potential in one cell to cross the cell membranes to generate an action potential in a neighboring cell. So what we're saying is that if we think about an action potential, what we have are ions that are traveling down the length of an axon. That's the action potential that's traveling. And so instead of generating an entirely new action potential in every cell, gap junctions are like tunnels that allow those ions to simply go through the tunnel and continue on to the next cell. So it's a way to connect neighboring cells together so that the original action potential can continue to flow through all of those neighboring connected cells. So we want that in places where we want there to be synchronous contraction, like where the timing of the action potentials is really critical. So like in the heart is a really good example of that. We want the atria, the chambers on top, to contract together, and then to have the ventricles, the chambers on the bottom, to contract together. So all of the cells on top are connected by gap junctions into one network so that when we have an action potential, we cause all of those cells to contract together, those cardiac muscle cells. And then that's separate from the network that's formed in the ventricles that causes those to all contract together, but after the atria contract. Okay, so that's an example of gap junctions. So we don't have them nearly as often in the body. Again, only 10% of the synapses in the body. And that's because there aren't as that many places where we want that kind of synchronicity. In most cases, we really want to have more fine control. We want to be able to cause action potentials independently, not necessarily cause one action potential that then travels through a whole piece of tissue. So we only have these in the heart, some areas of the intestines, and some areas of the brain. Okay, so they're relatively rare. They're faster than chemical synapses and allow for many cells to generate action potentials nearly simultaneously, rather than waiting the time it takes for a chemical synapse to occur at all of those different cells. Okay, so that is all I have for you, and I'll see you for the next video.